On this week's Arabidopsis Research Roundup, I'm absolutely delighted to be joined by Dr. Veronica Grieneisen from the John Innes Centre, and we are going to talk about a paper just published in PNAS that she's a co-author of, and the title of the paper is Auxin minimum triggers the developmental switch from cell division to cell differentiation in the Arabidopsis root. So this kind of follows on from work that has been done with collaborators, which looked at the Auxin maximum, and a lot of people who are interested in Auxin biology and root biology will know about the maximum, but this, as I said, is about the minimum, so we're really looking forward to hearing what Veronica has to say about this paper. So, hello, Veronica, and it'll be great if you could give us an overview of what you found in this paper.
Oh, yeah, well, thank you for having me here. So, in this paper, indeed, as you said, we discovered the partner of the maximum, so to say, which is the minimum.
Yeah.
And I think it was quite interesting because minimums are very much overlooked. And what we basically were looking at, so, in the lab, we're obsessed with this concept of positional information. Mm-hmm. So how do cells in an organ, any organ actually, know where they are and when to switch or change their behavior or their state? Yeah. So, for example, the root grows because it has stem cells, which are located at the Auxin maximum. Mm-hmm. So the Auxin maximum is very important. It's like the heart of the root.
Yeah.
After that, these daughters, of these stem cells, they will divide very quickly at the tip of the root. And at a certain position, they change behavior. So from one cell to another, if you look, you see that from dividing, they go to differentiating. So they stop dividing and they start maturing.
Yeah, okay.
And they will expand. And that's actually what gives that force to drive the expansion force to drive the root into the soil. Mm-hmm. So knowing where you have to switch your behavior along the root is very important for the root to grow quicker or more slowly. Mm-hmm. Now, the question was, okay, some years ago, I had modeling work which showed that associated with the quiescent center is a gradient. So you have to think about it like the apex of a mountain, and this mountain, having a certain slope and becoming lower and lower and lower as you go away from the tip.
Okay.
So one of the things people could think about from classical information theory and positional information is that at a certain amount or a certain level of that profile, cells would know when to change. Other people also propose that it might be with other genes, or it might have to do with the age of the cells and so forth. Mm-hmm. However, the work of my collaborator, Sabrina from Rome, she showed, and this has been known also for quite some time, that there is a tug of war between hormones going on. So cytokinin, which is another hormone, it has a tendency, if you give the root more of it, or there's more signaling of it, to make that meristem smaller. And if there's less of it, to make it bigger. So somehow these hormones were talking to each other. Mm-hmm. But now from a biophysics perspective, which is where I come from, my question is, what's the mechanism by which this other, hormone is actually able to influence auxin? Mm-hmm. And what we discovered in a nutshell is that through the way that cytokinin changes the pins, which are proteins that distribute auxin around, and in the way that cytokinin is also able to influence the degradation of auxin, it warps this gradient and it forms a valley, so that there's a minimum appears. Okay. So at the very tip, you have high concentrations that gets lower, it goes to a dip, to a minimum, and then it starts rising again. Mm-hmm. And so now what we hypothesized was, well, maybe this minimum is important for this switch, for cells to know that they should change from dividing to differentiating.
Okay. So, as I said, the paper is really a mix of physiology and genetics and also some modelling work as well. So you talked about this Auxin minimum. So were you able to find, maybe you didn't do this work, but in Sabrina's lab, were they able to find actually the molecular partners which played a role within this, defining this minimum?
Yeah. So they already had molecular partners that they knew were very important, but they didn't know how they were mechanistically influencing this transition zone. Mm-hmm. So the model at first hand, we were we're looking at it and putting more details of what cytokinin is doing. So cytokinin changes the pin distributions in the more proximal portions of the root. So when we implemented that, we realized that concentrations went back up. And then we also realized that degradation would be very important to locate this minimum.
Right.
Then they looked back, and together also with collaborators in the United States, Ross Lozani and Philip Benfi, they were actually able to see that there's a family downstream of cytokinin, the GH3 family, which is responsible for auxin degradation.
Right.
Now, modeling-wise, what we could confirm, and this was very surprising, degradation does not have to happen at the location of the minimum. It can happen somewhere else, but the minimum will appear always at the transition zone. So this is something which is very counterintuitive, and which really the modeling, you need the spatial modeling, right, to be able to understand that.
Yeah.
Now, the other thing is the modeling showed us there was a minimum, but we hadn't seen it yet experimentally.
Okay.
So that allowed Sabrina and actually Ricardo, which was the postdoc, which was leading this work, the lab, he then was able to very carefully look with different auxin reporters, and he could indeed behold that there was a minimum located at that transition.
Okay.
So at that position where cells transit from dividing to differentiating. Another student, which had been a long time in my lab also learning modeling, Nicole DeRuvo, she did the analysis on this raw data, and because you don't want to do it just by eye, you really want to quantify this.
Yeah, absolutely.
But the beautiful thing is when we looked at the mutants then, so mutants which would position maybe this minimum more proximally or more distally, so either making the meristem smaller or bigger, we always saw the minimum first moving and thereafter the meristem zone.
Right, okay.
So we could then, with many, many different instances and versions of mutants or overexpression or auxin treatments or cytokinin treatments, we could confirm that our hypothesis always was holding up.
Yeah, so you show that there's like an 8-hour lag between the position of the minimum and then the position of the zone.
Yeah, exactly. Because at first instance, you observe something in the model, and then you think, wow, this is interesting. Then you observe it, you make a hypothesis. Then you observe it to be true in the wild type.
Yeah.
And then you do all the perturbations. And then we can start saying, yes, there's a causal link, right? It's developmentally instructive. It's not just there, but it's developmentally instructive. Yeah.
Yeah, I see. So the model focuses very much on the GH3, the role of the GH3, so cytokine is upstream. It signals through a partner, the ARR1 protein.
Yes.
So have you only focused on the ARR1, or have you thought about other genes that might be involved in the cytokinin signaling and how they might impact, the position of the minimum, or how the root develops?
So that's extremely good question. At the moment, we have only focused on what's happening downstream of ARR1.
Okay.
But which is quite a lot, because downstream of ARR1, we have, through SHI2, we have pins, right?
Yeah.
And these pins are then being actually, they're being repressed and changing a little bit their localization. And so mechanistically, what I can say is that changing the strength of your pins at certain locations is able then to distort your auxin profile in a way which generates a minimum.
Okay.
And by augmentating degradation, which is also what's happening when cytokinin signaling kicks in, makes this minimum, this dip, very, very deep.
Right.
Okay. And so we think that this deepness, the deepness and the local confinedness of this minimum is very important.
Okay.
Because a cell which would transit through it, it would have to, in a short period of time, experience high concentrations, low, and then high again.
Okay.
And this is something which my lab wants to look into the future, because we are very interested in how do morphogen gradients get read out robustly. And so what we propose is now maybe these second derivatives. So second derivative means a minimum or maybe they are even more important than just absolute concentration values.
Right. Because
if absolute concentration values over the development of the root change, so they fluctuate or go up and down, the minimum will be resilient to it, because cells will still be able to experience a relative difference.
Yeah.
High, low, high.
Mm-hmm. Yeah.
And that's something that we of course want to, we have to look into through models in which we will actually be introducing growth.
Okay, so I would intuitively that might add a little more subtlety and the possibility of growth within that system, not relying only on a certain amount, but the relationship between an amount, two different amounts, and things like that.
So it's a qualitatively different way of how one processes morphogen information. Okay. Yes.
Okay, so thank you very much, Veronica. So it's been really interesting. I think we could probably chat for many, many more minutes, hours on this paper perhaps, but we'll wrap it up now. But I absolutely encourage people to take a look at this paper. I think oftentimes if you come from a molecular biology background, you see models and you get a little scared by them, but the way they presented it in this paper is really nicely done and very intuitive and very easy to follow. So I encourage everyone to have a look at the paper.
Thank you very much. And if I might just say a final word, by no means are we saying that this is the final story for how growth is happening. And I think there'll be many different hormones feeding into these mechanisms or mechanisms similar to that. Mm-hmm. So it's actually just a prelude to opening the doors to different genes, different pathways, and different hormones.
Yeah, absolutely. I mean, you mentioned within the discussion here there's been a new paradigm about Auxin signaling, which comes from the lateral root cap, which really isn't touched on here. And so integrating that into this, integrating all the different signals into the model.
Exactly.
Is really
will be very exciting.
Exactly.
Yes.
Okay, well, thank you very much, Veronica, for chatting to us today.
Thank you, everybody, for hearing.